I'd say in general, our laboratory is interested in gaining a deeper understanding about fungal pathogenesis in general, but also trying to build a better understanding about how human pathogenic fungi respond to antifungal drugs that we use to treat the diseases that they cause. We specifically focus on a fungal organism that is called Aspergillus Fumigatus. This is a mold type organism that is found pretty much everywhere in the environment. It's a ubiquitous organism. The current estimates are that all of us breathe in a couple hundred spores of this mold every single day. Whenever we inhale those spores, they're very small. They get way down into the lung. If you're healthy, your body is able to get rid of those spores completely with no problem. The patients who are at risk for developing an active infection because of those spores are people who are very severely immune suppressed. For instance, an individual who is having an organ transplant. What we want to be able to do in the very end is to develop new drugs that we can use to help cure patients who have these diseases, or to make the drugs that we already have better at doing that same job. And of course, we have some really effective antifungals that we use to treat patients who are diagnosed with these invasive fungal diseases, for instance, with Aspergillus. The problem is those antifungals are somewhat toxic to the host. We are also seeing increased rates of antifungal resistance with Aspergillus. And so we're trying to understand how that resistance develops and maybe what we can do about it so we can continue to use those drugs to treat patients who end up with these invasive fungal infections. The type of pathogen, the type of microbial organism that we study is what we call a eukaryote. The way that the fungal cells function is very similar to the way that our body's cells function. And so you can imagine then if I want to try to generate a drug that would specifically stop that fungus from growing inside of a patient's body, it's very likely that that drug is not only going to have an effect on the fungus, but it's also potentially going to have an effect on the host. And that's not what we want, because that's where we start to see problems with toxicity of a drug, because you don't want to impact the host other than to stop the microbe from growing inside the host. And for fungal infections in general, that's one of the biggest struggles. It makes it very hard to study these organisms. We know that there's something special about resistance because Aspergillus is everywhere in the environment. We don't have very many classes of drugs that we can use to cure these diseases. And when you develop resistance to one of those drugs within a class, oftentimes you lose the entire class of drugs. So we're really trying to sort of push forward our understanding of mechanisms that develop to sort of drive antifungal resistance, with the hope of understanding how to subvert that resistance and sort of save those antifungals so that we can continue to use them clinically.